Hello. I would firstly like to thank all of those directly involved in this work, including the EPSRC and Innovate UK for their funding, and UPC Cambridge for providing additional material. The current method of developing PMDI devices to match customers' performance requirements is iterative. Typical past developments could take several loops to get to design freeze for manufacture. There has often been minimal predictive simulation used in device design. The aim of our modelling work is to reduce the number of iterations by approximately 50%, thus significantly reducing development time whilst improving aerosol performance. For both pharmaceutical customers and their patients, this will shorten the time to market. This is a cutaway view of a PMDI valve, canister, and actuator. Spray area specifications at 30 mm and 60 mm from the actuator mouthpiece must be met for regulatory approval. These device and formulation variables can affect spray performance and were all variables in this study. A further metric of significance is the residual particle size distribution, or the impactor stage performance. This is linked to the droplet size distribution generated by the device, and the correlation between these is being explored in a current study at Recipharm. The emitted droplets undergo evaporation post-actuator. Size and morphology may change as a result of this. 4 device input variables were used in the current study: metering chamber volume, sump volume, spray orifice diameter, and spray orifice or land length. The propellant type and ethanol fraction were varied for the formulation. Spray area and droplet size distribution were the recorded output variables and were measured with the Preveris spray view or Sympatec key loss, respectively. In a conventional experimental study, each input variable would normally be tested at 2 levels in the first round of experiments. For example, the metering chamber volume might be set at 25 microliters for the lower level and 63 microliters for the upper level. In the first experiment, all of the variables might be at the second or upper level. For the second experiment, one of the variable levels will be changed, leaving all others as before. This process is repeated until all possible permutations have been exhausted. In the present study, with 2 levels and 6 variables, this requires 36 permutations. As the response variables could not be captured simultaneously, 72 experiments would be needed. For data collection in triplicate, 216 experiments would be required in total. Using a design of experiments approach, in this case a Taguchi orthogonal array, we can drastically reduce the number of required permutations while still ensuring that the results are statistically significant. In summary, the device and formulation characteristics on the left interact to form the outputs on the right. Partial least squares regression was used to analyse our results. This allows the relationships between the input variables and the response variable or response variables to be quantitatively described. Here we use the Nipal's algorithm. The response variable lies between the 2 ellipses and is shown here in green. Input variables lying between these ellipses explain most of the variance seen in the system. Variables inside the inner ellipse are less significant, but are not unimportant. The volume mean diameter and spray orifice diameter are located within the same quadrant and between the 2 ellipses. These 2 variables are therefore strongly positively correlated. Increasing the spray orifice diameter would therefore be expected to increase the volume mean diameter. Ethanol fraction and volume mean diameter are in adjacent quadrants, but still between the two ellipses. Here, they are negatively correlated across factor 1, but positively so across factor 2. As factor 1 explains 65% of the variance in the system, whereas factor 2 only explains 11%, we may take the behaviour observed across factor 1 as being representative. The remaining variables exert an influence on the response variable, but their influence is lower than that of the actuator orifice diameter or that of the ethanol fraction. The metering chamber volume is strongly positively correlated with spray area at 30 mm from the actuator mouthpiece. Larger metering chambers therefore increase the perceived spray area. Sump volume and R134A propellant are both negatively correlated with the spray area. Therefore, larger sump volumes decrease the perceived spray area. The use of R134A gives rise to smaller spray areas than were observed with HFO 1234ZE. Formulation ethanol fraction and land length exerted a minimal influence on the spray area. However, the samples tested during the study did not exhibit a significant difference in land length. A further study with test samples exhibiting a larger range of land lengths is required to better understand the influence of this on spray area. Spray orifice diameter has a bigger influence, as evidenced by being further away from the centroid, but is less influential than the variables described in the previous slides. In this case, larger spray orifices are weakly positively correlated with increased spray areas. This is the analysis for spray area as measured at 60 mm from the actuator mouthpiece for R134A only. The variable influences are similar here, but with spray orifice diameter being a much more significant contributor to spray area than at a 30 mm distance from the actuator mouthpiece. Partial least squares regression generates a linear equation for the response variable in terms of the input variables. The resulting regression coefficients generated by PLSR may be multiplied by the quantified normalized device characteristics and summed to yield the response variable. These equations may therefore be used to predict device performance. In this fictitious example, spray orifice diameter and ethanol fraction have the biggest influence on the response variable. The coefficients will depend on the normalizing factors applied to each variable prior to performing the PLSR analysis. For our next phase of work, correlating active pack droplet sizes to impactor data will add a further dimension to our modelling study, which is currently undergoing validation with placebo results. We would like to increase the scope of our study to additional low global warming potential, or GWP, propellants. Knowing the sensitivity of our response variables to the input variables, we can tailor future studies to increase predictive accuracy. For example, we could increase the range of the spray orifice length, or land length, to investigate the influence of this on spray area. In summary, we aim to reduce the development time of new PMDI products by approximately 50%. Spray area and impactor stage performance must meet the regulatory requirements. A TAGUCHI orthogonal array was constructed as part of a lean testing approach. Spray orifice diameter and ethanol fraction were the strongest influencers of volume mean diameter. Spray area was strongly influenced by most device variables. Droplet sizes are to be correlated with impactor stage performance in a further study at Recipharm. Thank you for listening.